Hi, everybody. Dr. Stewart here. I'm going to do your lab for this week here in my office at home. I have the models with me, the same models that we would use in the lab at school. Now, the lab practical, it will be your last lab practical, comes up in 2 weeks, is going to include the material from today, which is all the structures of the renal and urinary system. It's going to be, I'll do a demonstration lab on urinalysis. I'll demonstrate some urinalysis techniques and weighing of the urine to see what its specific gravity is. Those things can be on the practical as well. That's one of those kind of labs that we would do together if we were allowed, but I'll show you how that's done. And then next week, I'll also do the reproductive system, just like I'm doing this one today, all the parts of the reproductive system. So the renal and urinary parts, the reproductive parts, and urinalysis concepts will be on that lab practical, and it will be 2 weeks from now. It'll be the week after the reproductive system. That will be the last of your lab practicals, and we'll finish up your lab requirements for the semester. Again, I will be asking you to watch these lectures, to write them up. For this one, you can go through your lab manual and just carefully fill out every one of the structures on your lab manual and take some pictures and send those to me. For the reproductive, and for the reproductive lab too. For the urinalysis lab, I'll ask you to take some of the data down and answer some questions about that. Take some pictures of that and give it to me as well. And so that will give you the your credit for the lab attendance and participation for this week and next week. So once again, we have a sheet. And this one, it's on your canvas. It has the kidney parts on it, and it has the reproductive parts on it. So both lists will be on the test. It's about the same number total as we had for the last practicals. You know, there are quite a few parts, but I think you'll get them relatively quickly. But anyway, you can, as you go through the lab manual and write down every single part that they want you to write down, highlight the ones that are on this sheet so that you'll know which ones to concentrate on, to focus on more, or if you don't highlight them, at least keep this handy when you're studying for that lab practical, so you'll know the ones that I'm most likely to put on the practical exam. All right, so to start off with the structures, first of all, we have a kidney, and it is the convex shape out here, this dimple in here that has all of the structures coming in and out of it, is the hilum, the renal hilum. And so from the outside, you're seeing the hilum where all of the parts go in. If you open up the kidney and get inside the hilum area here, you will see the renal artery come in, the renal vein, the blue one. Here's the ureter going out, taking the urine out to the body. Inside here, this whole open area in here that contains all of these structures is called the renal sinus. And this is correct. I may have said it slightly differently in the lecture when I was pointing it out. The whole area is the renal sinus. The opening in here where all the urine drains into is called the renal pelvis. The renal pelvis is what accepts all of the actual urine, but the renal sinus includes these structures that are going to collect that urine and steer it into the renal pelvis. The outside of the kidney is the cortex, all the way around. That's the cortex. It has its own set of structures in it. The inside area is made up of these pyramids. There are several pyramids inside the kidney, and they all together make up the renal medulla. So the medulla is the pyramids. All of the pyramids together make up the medulla. The cortex is on the outside. So this pyramid, shaped like a pyramid, okay? On the tip of the pyramid here, all of the urine is coming from the nephrons, which are way out in here. It drains down into here and comes out this pointy end, which has a whole bunch of holes in the end, and that's a papilla. The papilla drains out the urine into this little funnel-shaped structure here called a calyx, C-A-L-Y-X. You have a calyx coming off of each papilla. When a couple of calyces come together, you have a major calyx. These individual ones are minor calyces. They dump into major calyces, which then dump into the renal pelvis. See what I've got going here. Okay, let's switch to the other model. So this one is cut a little bit differently. Cortex is all the way around the outside. The pyramids make up the renal medulla. You have the minor calyces receiving the urine from the papilla. They join together and become major calyces, which then dump into the renal pelvis, and then it steers the urine down into the ureter, which is going to go all the way down to the bladder. So coming in here, we have the renal artery. It goes out into these interlobar arteries, the interlobar arteries, kind of between these lobe-like structures. They branch off into the arcuate arteries, which look like arches. And then off the arcuate arteries come the smaller vessels, which again, your manual may call interlobar arteries, but I'm calling these interlobar arteries interlobar arteries. The ones out here are going to be your arterioles that are going to the nephrons themselves. Here we have a closer up version. So this whole structure here is a nephron. We have the glomerulus, and we have the proximal tubule and the distal tubule and the collecting duct. The afferent arteriole is going to come in from this artery, okay? So if I ask you on the test, if I point to this little guy, if it's coming from the artery, it's the afferent. It's coming in. The efferent artery goes out and then goes into these peritubular capillaries that go around the tubules and start taking up the fluid. You also have some of those down here. It doesn't show well on this, but I'll show it to you on a different one. These little berries from the outside, they call them renal corpuscles. A renal corpuscle is the entire structure from the outside. Looks like a berry from the outside. Once you break it open, though, then you can see the individual structures of it. You have Bowman's capsule. You have the glomerular capillaries inside here, afferent coming in, the efferent going out. You have the proximal tubule leaving the glomerulus here. The glomerular filtration goes down the proximal tubule and becomes your primary urine. So if it's leaving there, there's your proximal tubule, the descending limb, the ascending limb. Then the ascending limb comes right back next to the glomerulus here. See, here's the proximal tubule. It's carrying the urine away. Proximal tubule carries it away. It goes down and around and comes right back next to the glomerulus itself. And here is your macula densa. The macula densa is that set, so it would be right there on this one. the macula densa, right where that distal tubule comes back and touches the glomerulus, you have a group of sensory cells that tell you what's going on and therefore can secrete vasodilators and vasoconstrictors to increase or decrease the action of the nephron. So this is your tubuloglomerular feedback. From the tubules comes the feedback to the glomerulus, and therefore you're going to constrict or dilate. Tubuloglomerular feedback. You're going to constrict or dilate to control the action of that nephron. Remember, you have the proximal tubule. I'll actually use this other model. It's bigger, and it'll probably be used on the test anyway. So here you have some of those renal corpuscles. Okay, the afferent arteriole is the one that's coming in from the artery. The efferent goes out, so afferent comes in. The efferent goes out around these peritubular capillaries then that allow you to take back some of that fluid. And you'll see there's some way down here around the loops of Henle too, because you're going to have to reabsorb here. You're going to have to reabsorb here. So you filter in the glomerulus. The P goes down the descending loop of Henle, ascending loop of Henle, and you have these peritubular capillaries at the bottom and up here, and then at the end of this ascending loop of Henle, first of all, you go past the glomerulus to have the macula densa to signal it, but then you have the distal tubule, which dumps in, here we go, that's a good one. There's the distal tubule that dumps into the collecting duct. The big yellow thing is the collecting duct. So we're going to have a proximal tubule going in. We have the descending loop of Henle, ascending loop of Henle, and then it comes back close to the glomerulus at the macula densa. It becomes the distal tubule, and it dumps into the collecting duct. Okay? Dumps into the collecting duct. See, in this case, it's coming out the top. So descending loop, ascending loop, macula densa is here in this one. Distal tubule is connected to the collecting duct. So it's easy to remember the proximal tubule is connected to the Bowman's capsule. The distal tubule is connected to the collecting duct. So, although I'm looking at it upside down and backwards, you ought to be able to tell, if I give you a really good close-up picture, which one is which. If it's coming out of the glomerulus, it is the proximal tubule. If it's going into the collecting duct, it's the distal tubule. Now you have a group of these red and blue things are not tubules. The tubules are connected to the glomerulus and the collecting duct. These blood vessels down here, it's a group of straight up and down blood vessels, which, if you remember, rectus abdominis goes straight up and down, rectus femoris goes straight up and down the front of your leg. This is called the vasa recta. Straight up and down blood vessels is what it means. So there's an area of blood vessels here that is going to supply all of these peritubular capillaries. They're going to give the blood and then take it back with the blood in it and then take it back to the body. So the vasa recta are these blood vessels outside the tubules, the kidney tubules, that are supplying the blood out here. Straight up and down blood vessels are the vasa recta. Here we have a look at it on a picture that I might also use. Okay, so here we have the And I would give you an arrow, of direction for this. If the blood's coming in here, see this little white arrow? I'll make it clear. If it's coming in this way, this is the afferent arteriole. It goes back out the efferent arteriole. The glomerular capillaries in here, inside of Bowman's capsule, filter. That becomes the primary urine. It goes all the way down the tubule. It goes down the descending loop, the ascending loop, comes back right by here. Here again is your macula densa. It means dense plaque. And there's a little group of cells here. They're usually a little darker so it looks like a plaque of sensory cells. And then it goes and dumps into the collecting duct, where, as you know, a lot of fine tuning takes place with all those principal cells in there. All right, so on this one, a more general look at it, you have your renal artery coming in. So this is the whole kidney. There's the adrenal glands on top there. Here's the hilum, okay? Inside you have the sinus, this whole area where you have the papilla and the calyces dumping into the renal pelvis, which dumps into the ureter. Here they have these tubes on here. These are the ureters, which go down to the bladder. The bladder sits right on your pelvic floor, so it's as low down as it can get on the pelvic floor. The ureters take the urine all the way down to the bladder, and they take them in sort of the lower back. They come up in the back and slightly inferior part of the bladder. They don't go down in the top. They kind of go up in the bottom. And so what you have when you get into the bladder, here's a female bladder. If I can get it to focus decently. You have the ureters coming down, and you can see two little holes here where the ureters dump in. Two little holes here and here. Maybe show it better with a pen. Okay. Here we have here and here. And then you have the urethral opening, and the three of those together form a triangle that's called the trigone region. Same in the male. We have the two innies from the two ureters, and we have the outie, which is the urethral opening. That's the trigone region. It's an area that is highly susceptible to bladder infections because you have stuff coming in and stuff going out there, and things can collect there and cause bladder infections. The big muscle surrounding the whole bladder is called the detrusor muscle. When you want to pee, you have to relax the sphincters and contract the detrusor muscle, so it squeezes it out. When you don't want to pee, you relax the detrusor and you squeeze the sphincters so that no pee comes out. Parasympathetic causes you to pee. Peeing is parasympathetic. Sympathetic inhibits it. So while you're running, your detrusor relaxes and your sphincters contract. When you're at rest and it's time to pee, the detrusor contracts and the sphincters relax. Big difference on the male and female here, and we'll see this even more in the reproductive, is the male has this prostate gland. The prostate gland. And the urethra goes right through the middle of the prostate gland. It's why older guys that get enlarged prostates have a hard time peeing. It kind of squashes off the urethra. This is the part of the urethra that's going through the prostate. The internal sphincter, urinary sphincter, is right before the prostate. It's right at the bottom of the bladder. That's the internal sphincter. The external sphincter is a ring of muscles way down here beyond the prostate, right at the pelvic floor. The internal sphincter is autonomically controlled. The external sphincter is controlled voluntarily, just like the anal sphincters. In the female, you have the internal sphincter right at the exit from the bladder. It is automatically controlled. The external sphincter, you see these muscles, they look a lot like the anal sphincter muscles. It's a ring that goes all the way around right at the pelvic floor. Internal and external sphincters to control urination. As you can see, the urethra on the female is like 1.5 inches long. That makes it really easy to catheterize. If a female needs a catheter, it's a lot easier than on a male, but it's much easier to get urinary tract infections because it's so close. The opening here is close to the anus, and the vaginal opening is easy to get infection in here, whereas on the male, the opening is 3 feet away. The male would like you to think, actually a few inches farther away. And so it's a lot more difficult for any bacteria or something to get from this perineal region into the tip of the penis to come all the way up here. So the male gets fewer urinary tract infections, but it's much more painful to catheterize a male if you need to. And the last structure on here is the urethral orifice. It's the tip where the pee comes out. It's also called the urinary meatus. A meatus is an opening, if you remember the auditory or acoustic meatus, that your ear canal goes through. So this is called the urinary meatus or the urinary orifice. On the female, same thing. It's right where the pee comes out. If I can get it over there, my handle's in the wrong place. Here, the urinary orifice is right at the tip. Okay, so those are the structures of the urinary system I'm going to want you to know. I'll add about the same number of structures for the reproductive system, and those will be on your lab practical.